seminars, special lectures of various types. But one of these annually, uniquely, is titled The UCLA Lecture in the Sciences. For this lecture, we bring to the campus a man who is both eminently distinguished by his contributions to some area of science and noted for his ability to make this field meaningful to those of us who aren't specialists in his field. This year, we're very fortunate to have a man who superlatively fills both of these criteria, Sir Vincent Wigglesworth, who has for the last 2 years been busily divesting himself of various administrative responsibilities so that he can turn, I think, full-time to research again. He retired a year ago as professor of biology at Cambridge, and just this summer as director of the Agricultural Research Council unit in insect physiology. Because of the enormous economic and medical importance of insects, there's been a tendency, at least it seems to those of us outside the field, for the applied aspects of entomology to tend somewhat to overshadow, in many cases, fundamental science. At least in the early years of the development of the science, this was so. In thinking of early fundamental work, we might think of Morgan's group working with fruit flies. But I think it's almost fair to say that Morgan was interested in the fruit fly only as a rather odd package that nature happened to have devised for containing Drosophila chromosomes. Sir Vincent was one of the small group of pioneers who made the study of insects a scientific study in its own right, especially all aspects of physiology and development of insects. It's hard, in fact, to find a subdivision in this large field in which he hasn't made major contributions. In the course of his study of development, he founded, as everybody here knows, the subject of insect endocrinology, in finding the hormones whose balance determines, or at least in large part determines, the course of development. His textbook, The Principles of Insect Physiology, for nearly 30 years has both, in its successive editions, chronicled developments in this field and guided the development of the field of insect physiology. In addition, he's been for many years noted for the lucidity of his reviews and his popular and semi-popular articles and lectures. We're very glad to have you tonight, Sir Vincent. His topic is preformation and insect growth.
Well, thank you very much indeed, Professor Atkinson, for these very kind words in the introduction. It is a very great pleasure for me to be invited to give this annual science lecture, and an honor which I appreciate very much indeed. It is more than 22 years, I think, since I visited Southern California, and during that time, UCLA, to use a slight understatement, seems to have become transformed. And it is very exciting for me to be invited to come back to the campus of this dynamic university to give this lecture. I understand that what I'm supposed to be doing is to giving a lecture which is not only intended to be satisfactory for experts who want up-to-the-minute account of the problems, but also to the general public. I suppose this is the sort of the sort of assignment which this university makes to its faculty. But it is the sort of impossible assignment that I'm afraid I am unable to fulfill. But I will do the best I can. Now, what I propose in this lecture is to take as my starting point the famous Dutch naturalist Jan Swammerdam. There are many discoveries of fundamental interest to the insect physiologist in the pages of the Biblia Naturae or Historia Generalis Insectorum of Swammerdam. But I shall concern myself particularly with Swammerdam's contribution to the understanding of growth and metamorphosis. The scientific study of insect metamorphosis, of course, goes back to Aristotle, who first put forward the idea that the insect larva, these are what he wrote, that the insect larva, while it is yet in growth, is a soft egg. In other words, that the embryonic life of insects really continues right up until the formation of the adult, the perfect insect or imago. Some many centuries later, William Harvey, in his second great work, the Generatione Animalium, published in 1651, extended his interests beyond the mammals and the vertebrates and devoted much attention to the generation of invertebrates and particularly of insects. We do not know to what extent Harvey's conclusions on this subject were based upon his own observations, for unfortunately all his records have been lost. But extending the argument of Aristotle, Harvey developed the theory that the insect egg contains so little yolk that the embryo is forced to leave it before completing its development. And it then goes through a more or less prolonged larval stage, or succession of larval stages, during which it builds up food reserves, stores up food reserves, before resuming the egg stage, the egg form or pupa, and completing its embryonic growth. Harvey recognized, quite rightly, that the substance of the full-grown larva is the material, furnishes the material for the production of the adult insect. But he supposed that under the action of some mysterious influence, this material suffered a metamorphosis and was rebuilt in an entirely new form, like wax bearing the impress of a seal, as he wrote. It was this miraculous element in Harvey's conception of insect metamorphosis that Swammerdam was out to challenge. The Biblia Naturae was written in 1669, some 18 years after Harvey's work. And in his courteous refutation of some of the opinions of what he recalls this great philosopher, he excused the mistakes of Harvey on the grounds that insects are difficult to study unless by persons accustomed to experiments of this kind. It is no wonder, he writes, that the most happy geniuses, the immortal Harvey, for example, and many others, should have fallen into an error. Could I have the lights and the first slide, please? For Swammerdam, epigenesis, a gradual and natural growth, is the only way in which all animal form is fashioned. He stripped away, in these classic experiments as illustrated here, he stripped away the skin of the caterpillar before it was cast off and found the wings and other parts of the adult butterfly already in process of growth. He inferred that every structure arises by a process of continuous growth from an existing structure, and that everything is already preformed in some state, some invisible state that we are unable to appreciate from the earliest stages of growth. As growth proceeds, the parts become visible and differentiated. We have at home a familiar invocation, "May heaven defend us from our friends." Unfortunately, the followers of Swammerdam adopted the crude and exaggerated idea that all the future structures of the body are actually literally preformed in a literal sense in the egg, and that all growth and development consisted in the unfolding of these structures as successive covering shells were discarded. The height of absurdity was reached when observers claimed to have seen homunculi, the diminutive form of diminutive outlines of the human form in the spermatozoa. It was excesses such as these which brought the very reasonable views of Swammerdam into disrepute. Swammerdam was trying to express ideas for which, as yet, there was no adequate foundation in physiology. One must realize that at the time he was writing, chemistry was virtually non-existent, cells were unknown, the foundations of histology were only just being laid by Malpighi, genetics lay 2.5 centuries ahead. The best idea that he could suggest was that the unformed organs were in a sort of dissolved state, and that the evaporation of water caused them gradually to separate out. Harvey's theory that the insect egg cannot complete its development because it is deficient in yolk was shown by Swammerdam to be untenable, because in the more primitive insects, such as the silverfish, which you see here, and in grasshoppers, the creature that emerges from the egg has the same general form as the adult. It is in the higher insects, such as beetles, as you see here, or butterflies and moths, in bees or flies, that the most striking examples of metamorphosis are found. And yet, the eggs of these higher insects are not notably more deficient in yolk. But when it comes to the understanding of metamorphosis, we are still very much in the dark. But the great advantage over Swammerdam that we enjoy is our knowledge of embryology and genetics. The insect begins its development as the fertilized egg or zygote. The chromosomes of the zygote carry, encoded within them, the blueprint for every structure and every function of the future organism. The zygote divides, and as the cleavage nuclei resulting from that division migrate to the periphery and enter the cortical plasma of the egg, in the mosaic type of egg that is, they become committed to form the different regions of the body. The cleavage cells that arrive in this zone form the head, in this zone the thorax, the abdomen, the gonads, and so on. We see here an example of polymorphism. The segmentation nuclei seem to be molded by the environment in which they find themselves. As they migrated through the yolk, they still retained the capacity to form any and every part of the organism. Now they can build only the structures characteristic of some particular region of the body. This progressive polymorphism of the parts of the body is, as you know, called differentiation. It seems that local chemical differences or gradients of one sort or another call forth particular elements or capabilities in the chromosomal blueprint. And when that has happened, there is generally no return. As Spemann has said, particular powers have been evoked, more generalized potency has been lost. We do not know precisely the nature of this chemical transformation. There is some evidence that the characteristic effects of these different regions of the cortical plasma, which these regions exert upon the nuclei which enter them, may depend upon differences in the nature of the RNA content of each zone. In these and other examples that I could quote, it is the cytoplasm which seems to be exerting the determining influence. It is for this reason that it has been customary in the past to ascribe to the cytoplasm the responsibility for differentiation. But in recent years, the tendency has been to put the onus for the developmental changes upon the genes in the nuclei, and to describe these cytoplasmic effects as controlling the activation of the relevant parts of the gene system. The development and differentiation of the embryo is an immensely complex matter. A single cell, the zygote, differentiates in a few days or even a few hours into a complex organism with sense cells and nervous system, muscles, alimentary canal, circulatory system, and the rest. The whole process is so bafflingly complex that it seems to defy physiological analysis. Excuse me. But as Swammerdam realized, embryonic development continues right up to the adult stage. What we need is to isolate a suitable part of this process, and so to provide a simpler system which will be more amenable for experimental study. Such a system is to be found in the epidermis of the insect. As was originally shown by Haeckel in the middle of the last century, the integument of the insect consists of a single layer of cells which lay down a more or less inert cuticle. Each cell is responsible for laying down its own tiny area of cuticle, which thus consists of a mosaic made up of the contributions of the thousands of individual epidermal cells. And now the epidermis is the chief agent in the control of the body form of the insect. The pattern of the cuticle reflects the differentiation of the cells below and provides a permanent record of that differentiation. Here we have a system in which we can observe differentiation at work. I propose to take a very simple example, the epidermis over the abdomen of the blood-sucking bug Rhodnius prolixus, which you see a fourth stage larva here. Over a small area of the surface, the integument consists of cubical or flattened epidermal cells with their overlying cuticle. And the obvious function of each epidermal cell is, as I've said, to secrete its own little area of cuticle. And that is an exceedingly complicated chemical and physiological process. In the resting state, that is, in the unfed, non-growing insect, this cell is not a very impressive object. It is exceedingly flattened and attenuated, not more than some 3 microns thick. The nucleolus is small. The scanty cytoplasm contains just traces of ribonucleic acid, a few scattered spheres of red pigment, and relatively few mitochondrial granules and filaments. It rests upon a basement membrane, but is not intimately attached to this. On its outer surface, it is adherent to the cuticle, which is penetrated by these fine canals, the so-called pore canals. And here is a photograph in surface view of these resting cells with their small nucleoli and granular and filamentous mitochondria. Within a few hours after feeding, these cells are activated. The nucleolus enlarges. This photograph was taken about rather less than 24 hours after feeding. The nucleolus, as I say, enlarges. The mitochondria become swollen and divide and break up into and elongate to form filaments, which break up, and so numbers of mitochondria increase rapidly. Ribonucleoprotein increases rapidly in the cytoplasm. Within 2 days, the epidermis forms a cubical epithelium. By this time, the cells are getting so thick that the view is, of course, rather hazy. The cytoplasm is filled with rapidly increasing mitochondria. Large amounts of RNA are now present. The nuclei are enlarged. The nucleoli are greatly enlarged and lobulated. Exceedingly active, obviously, exceedingly active synthesis, protein synthesis is in progress. By 4 days, cell division has begun. Here you see these attenuated cells in the unfed end. Here, I suppose, is at about 2 days. 4 days, you see cell division actively in process. And then, when mitosis is finished, the cells arrange themselves in proper form and begin to lay down the new cuticle. In Rodnius, only one single meal, one meal is needed for each mode, so the whole process is very readily observed and timed. Now, the subsequent activities of the epidermal cell during this process of cuticle formation is summarized in this slide. It first lays down this outermost layer, this thin layer of lipoprotein, which becomes the so-called cuticulin layer of the epicuticle. Then the micelles of chitin and of protein are laid down in successive laminae around the pore canals. When the new cuticle goes on, and when the new cuticle is nearing completion, these cells, the epidermal cell again, produce and discharges these enzymes, chitinase and proteinase, which are discharged through the substance of the cuticle, perhaps by the pore canals, and discharged into the space between the newly forming cuticle and the old cuticle, which is not represented here. These enzymes digest the inner layers of the old cuticle, and sometimes as much as 90% of the old cuticle may be dissolved. In this way, the products of digestion are reabsorbed through the new cuticle by the epidermal cell and are doubtless utilized in the formation of the new cuticle. About this time, another product of the epidermal cells appears on the surface of the epicuticle. That is a proteinaceous layer which actively reduces ammoniacal silver. And since that reduction is often believed to be due to phenolic substances, we often refer to this layer as the polyphenol layer. And this material seems to exude from the ends of the pore canals and to form a layer over this so-called cuticulin layer. It forms this thin, continuous layer. And shortly before molting, the epidermal cells gives rise to yet another substance, a very important one, which is the wax, which crystallizes out over the surface of this reducing layer, and highly important because that waterproofs the insect in readiness for casting off its old waterproof skin. And meanwhile, those regions of the chitin protein layer, which are going to be hard and horny in the next stage of the insect, become impregnated with more protein and with phenolic substances. And it is at this stage, about this stage, that the undigested remains, that is, only the most superficial layers of the old cuticle, are shed, and the insect escapes in its new cuticle. And then, within half an hour or so, the final layer of the cuticle, that is, this protective layer of cement, is poured out over the surface. Now, that alone, of all these other substances, is not produced by the epidermal cell. All this range of changes and enzymes and so on are all the products of the epidermal cell. The cement layer is, however, a product of dermal glands, whose ducts are passed between the cells, and discharge of this material is discharged over the surface. Now it remains only for the epidermal cell to secrete into the cuticle phenolic phenol oxidases, which oxidize these phenolic materials to quinones. These quinones tan the cuticle. The insect is said, in fact, to tan its own skin and convert these proteinaceous contents to the hard and horny substance which we call sclerotin, and which oxidize other phenolic substances, notably tyrosine and its derivatives, to the black pigment melanin. And now that is the completed process. Now we have the new cuticle complete. Now I've rather labored this story in order to emphasize that the epidermal cell is a functional cell with an amazing range of chemical and physiological activities. But at the same time, it is an embryonic cell. It contains a considerable capacity for differentiation. When the insect grows, it first lays down a new and larger cuticle, and then it molts and casts off this old cuticle, as I've just described. And during this process, a given epidermal cell may divide and retain its orderly properties and appearance, give rise to daughter cells with the same characters which secrete a new cuticle. But alternatively, this cell may divide into 4 daughter cells which become differentiated from one another. 2 of them become large cells, as you see here. Of these large cells, one is the so-called trichogen, which gives out a process which will become a sensory hair or bristle. And the other one grows around the neck of that bristle and produces this socket in which it is articulated. The third cell is this one, gives out a process which grows inward, and here is this process which grows inward, and another process which grows outwards to the base of the bristle. And the inward growing process is an axon, nerve axon, which grows onwards until it chances to meet another small nerve. It associates itself with that and follows it, and eventually, by those means, finds its way right to the central nervous system. So here we see a part of the central nervous system arising by differentiation from an ordinary epidermal cell and being added to the ganglion or brain or whatever it may be. The fourth cell, this one, is the so-called neurolemma cell, which provides a sheath for the sense cell and the distal process, and a sheath for the axon. In this way, we see the process by which an ordinary epidermal cell becomes differentiated into this little sense organ or sensillum. Well, how is this process of differentiation determined? Sensory hairs, each surrounded by a smooth dome-like area or plaque, as you see here, are scattered fairly evenly over the surface of the abdominal segments. And here you see half of one abdominal segment with with these plaques and sensory hairs scattered over it. And in the figure below, you see the same region of the abdominal integument after molting, and you see shaded in grey new plaques, new sensory organs which have become differentiated. And you can see that those new ones have appeared in the places where the gaps between the existing ones were the largest. It is therefore possible to a considerable extent to predict where these new ones are going to appear. They're never developed close to the site of an existing hair. Now, if the epidermal cells below an area of the cuticle are killed by touching the cuticle with a heated rod, the cells from the surrounding regions divide and migrate inwards, dividing as they go, and thus restore the continuous epidermal sheet. Such a burn has been inflicted here in a second stage larva, and here you see the resulting cuticle after healing when this insect has molted to the third stage, and you can see that the central area which has been healed does not carry any sensory hairs. But when this insect molts again, sensory hairs and plaques are formed over that area at the normal intervals. And when that insect molts again, the remaining gaps are filled in by further new plaques scattered at the regular intervals as in the normal insect. It thus seems that each sensory hair seems to exert an inhibitory influence around it, which prevents the appearance of a new hair in its immediate neighborhood. There are, of course, a number of possible explanations for this. The idea that I favor is that some chemical material in the cytoplasm of the epidermal cells is necessary for the development of a sensory hair. When a hair develops, it drains this hypothetical substance from the surrounding area, the surrounding cells, and thus prevents these cells from differentiating to form sense organs. But beyond a certain distance from the existing hairs, there are cells that are not deprived of this hypothetical substance. Development of a sensillum begins, and this in turn drains away the active substance from the surrounding zone. Well, what is the meaning of these observations? It would seem that there is a gradient of some kind around each little sensillum. Perhaps this is a concentration gradient of some chemical substance. When the substance exceeds a threshold level, the gene system of some epidermal cell is activated, and it begins to develop into a sensillum and drains off the active substance and thereby inhibits a similar development in the surrounding cells. This, of course, is a purely hypothetical conception, but it is one which explains the facts and is useful as a tool for thought. Now, the epidermal cells may also undergo another type of development. I won't describe this one in detail. There is one here in which it divides into two cells, you see, to give rise to these enormous cells, the so-called enocytes, which we believe are responsible for producing the material for the epicuticle. But what I want to call your attention to this other type, in which you again get 4 daughter cells, just as in the formation of a sensillum, but here they differentiate. One of them becomes a large cell which becomes vesiculated and forms a large sort of vesicle here with a thin cuticle around it, and in fact becomes a dermal gland. And the other 3 cells contribute to the formation of the duct and the sheath. And these are the dermal glands that produce the cement layer which is poured out over the surface. Now, these dermal glands are likewise scattered evenly, or fairly evenly, over the surface of the abdomen, as you see, but they are more closely spaced than the sensilla. And we saw in a slide just now that in the first molt, that takes place after burning an area of the epidermis, although no sensory hairs and plaques were formed, but as shown here, dermal glands are formed. This shows the integument immediately after molting, before the glands have discharged, so that they show up very conspicuously by their distended little vesicles. Now, here is the control side with the dermal glands, and you can see the hairs and plaques. This is on the side which has been burned. No hairs and plaques have differentiated, but dermal glands have differentiated at the normal, more or less the normal distance apart. And it may be that the same inducing substance that is necessary for the production of sensilla, when it is present at a lower threshold, will activate that part of the gene system which leads to the development of a dermal gland. Excuse me. This idea that an inducer substance evokes different characters which are latent in the gene system when it is present at different levels of concentration is an important one for understanding the nature of growth and differentiation. There is evidence that in each segment of the abdomen of the insect there is a similar gradient which is responsible for determining the polarity of the sensory hairs as they grow out. The new sensory hairs, as they appear, don't grow out in any undetermined direction. They always grow out in an anteroposterior to that direction. And you may have noticed that even where they appear over a newly healed area, you again get this normal orientation or polarity of the hairs. And a recent research student of mine, Peter Lawrence, has produced good evidence that this results from a chemical polarity in the anterior to the posterior end of each segment. And it has been shown in Germany by Marcus and recently confirmed by Stumpf that in the pupae of the wax moth, Galeria, each level along this gradient of the segment, each segment evokes a characteristic type of cuticle. And they showed that if a minute piece of the integument is removed from one part of this one point in this gradient to another point, it will develop characters which are determined by the new level of the gradient into which it is transplanted. Now that is a very important experiment which, if extended and confirmed in other groups, I think will prove to be a very important contribution to the understanding of differentiation. Of course, the epidermis of the abdomen, which we're using here as a model of embryonic growth, has already undergone a large amount of differentiation. In the larva of Rodnius, which you see here, this is the abdomen, the thorax, you see the wings, and the abdomen here of the 5th stage larva in Rodnius. Not only it is divided, as you see, into a series of segments with these intersegmental membranes between. And each segment also bears a pair of pigment spots, black pigment spots, rounded, oval pigment spots at the posterolateral angle of each segment. Characteristic patterns of the arrangement of sensory hairs and their plaques, and so forth. Now, what I want to call your attention to in the larva is that in the larva, these rounded oval pigment spots lie at the posterolateral angle of each segment, and that occurs throughout all the larval stages up to the 5th. When the 5th stage larva molts into the adult, you get this striking metamorphosis. And I say 5 of these larval stages, but on the 5th molts become the adult, you get this striking metamorphosis in which large membranous wings are formed, the genitalia are differentiated, and there is a complete change in the structure and pattern of the cuticle of the abdomen. Sensory hairs and plaques are absent now over the central area of the abdomen. They are present only along the sides. And there are very many other changes, but the one I particularly want to call your attention to is that the pigment spots are no longer oval and situated at the posterolateral angles. They are trapezoidal spots at the anterolateral angles. Now, we saw earlier that if the epidermis below the cuticle is killed by heat, the epidermal cells from the margins of the burn divide and migrate inwards until the deficiency is repaired. And as these cells grow inwards, they carry with them the characters for which they've been determined. So that if a burn was inflicted like this, you see, so that it just passed through the margin of these black pigment spots, as you see here, then when that wound heals and the insect molts again, you find that there has been a centripetal migration of those pigment spots in this way. Now, it is possible to inflict a burn of this sort between 2 pigment spots in a young larva. We'll say here in a 2nd or 3rd larval stage, and it's not surprising that when that molts, you see now you show it at a later stage, in the fifth larval stage, those two pigment spots have joined up. Or a pigment spot can be burned out like that, and then in the later larval stages that pigment spot is absent. But as we saw, these spots in the adult have quite the opposite distribution, and therefore when this insect becomes adult, you find that where the two pigment spots are united in the adult, what you have done is really you will now have joined up two areas which were destined to be colorless in the adult, so the pigment spots have disappeared. Conversely, where the pigment spot in the larva had been burned out, the pigment spots in the adult have become united. Swammerdam would have appreciated this experiment, for it illustrates his point extremely well. It shows that even in the larva, with its characteristic pattern of the body, the totally different pattern of the adult is already laid out, but in an invisible form. Cells actively engaged in forming some element of the larval pattern have already been determined to form some quite different element in the pattern of the adult. The adult pattern is quite clearly preformed. Now this brings us to the question of metamorphosis. Here you see the fifth stage larva of Rodnius with its abdominal cuticle as I've been describing it, and its rather stumpy wing lobes. When that molts, it develops into the adult, as you see here, with large membranous wings, with a cuticle which is totally different in structure and pattern, thorax which is completely transformed, ocelli have appeared for the first time behind the compound eyes, the external genitalia have become differentiated, and so forth. The more closely you look at this insect, the more impressive is the change in form which it has undergone. Well, what brings about this switch from the larva to the adult? We have known for many years that this is brought about by a change in the circulating hormones. The process of growth, which leads on to molting, is set going by a secretion from clumps of neurosecretory cells situated in the dorsum of the brain. The axons from these neurosecretory cells cross over and terminate in swollen endings in the corpus caldacum, this nervous body just behind the brain. There, the product is liberated as the so-called brain hormone, which acts upon another endocrine organ situated in the thorax, the so-called prothoracic gland, which is then activated and caused to liberate the molting hormone, or as it is called, ecdison, which acts directly on the epidermal cells, causing them to undergo this process of molting and cuticle formation. And immediately behind the corpus caldacum is another glandular organ, the so-called corpus allatum, which secretes a third hormone, the so-called juvenile hormone. In the presence, when the molting hormone acts in the presence of the juvenile hormone, then the epidermal cells lay down a cuticle of larval type, and the insect therefore retains its larval form and characters. And that happens throughout the first, second, third, and fourth larval stages. But in the fifth larval stage, the corpus allatum no longer secretes the juvenile hormone. The epidermis is therefore exposed only to the molting hormone, and under those circumstances, the epidermal cells change their activity, and they lay down cuticle of adult type, and the insect undergoes metamorphosis. if the corpus alatum is removed from a young insect and implanted into the abdomen of a fifth stage larva, you can see here the scar in the abdomen where implantation has taken place, then that fifth stage larva, instead of molting into an adult and undergoing metamorphosis, molts a grain into a characteristic larval form, with the type of cuticle, the wings, thorax, and everything else of the same type. Sometimes the experiment is not so successful as this. You get intermediate forms or stages between this giant six-stage larva and an adult. And you may get a perfectly good adult. As you see here, here is the larval abdomen of a perfectly normal adult with just a tiny little patch. You see the larval type of cuticle with these plaques and sensory hairs, which marks the point overlying the implanted corpus alatum. And if you section through such an area, here is the thin adult cuticle, the thin adult cuticle. Here is the thickened cuticle of larval type with its plaques and hairs. And here you see the implanted corpus alatum, which has exerted its action, juvenile hormone action, over this area. Clearly, the juvenile hormone acts directly on the epidermal cells themselves. Some 10 years ago, 1956, when Carol Williams was working in our laboratory, he found that the lipid extracted from the adult males of the Cecropia silk moth is an active source of the juvenile hormone. This was a very remarkable and a very valuable discovery, which for some odd reason it does not happen in other insects, but in this particular moth you get large amounts of the juvenile hormone seems to accumulate in the stored lipid in the fat body. And this material can readily be extracted by ether, and Dr. Williams very kindly gave me a sample of this, and it can be applied locally to the integument. For instance, it can be applied to one wing of a fifth stage larva in Rodnius, and you get the insect turning into a perfectly good adult with one larval wing. Or it may be applied to the abdomen, one segment of the abdomen, as you see here, and you have a perfectly good adult with a larval segment interposed with its characteristic cuticle and plaques, its characteristic pigment spot. One little point here you see has escaped the hormone. It's got a little tiny patch of adult cuticle there. It's even possible to engrave the abdomen of the adult with initials in this way, so you can recognize them. Now we saw that there is some evidence that graded concentrations of inductor substances can lead to qualitative differences in the characters produced. And the same is true of the juvenile hormone. In such insects as Lepidoptera, in which there is a pupal stage between the larva and the adult, it seems highly probable, I think the evidence is really good, that, as suggested by Pifo, the larval form is produced when the cells are exposed to the molting hormone in the presence of a large amount of juvenile hormone. The pupal stage is formed in the presence of molting hormone with only a trace of juvenile hormone, and the adult is formed in the presence of the molting hormone alone. Now these morphological effects are spectacular, and of course the details of the effect are quite different in each part of the body. And yet the whole transformation is can be brought about by a single simple chemical compound. Materials with juvenile hormone activity are widespread in nature, as was shown particularly by Schneiderman and his colleagues. They can be extracted from a wide range of invertebrates, from the tissues of vertebrates, including man, from many protozoa, bacteria, and plants and yeasts. In 1958, Peter Schmalek examined two of these sources, the excreta of the mealworm, Embryo, and yeast. And he found that in both cases, the active principle appeared to be a mixture of the isoprenoid alcohol, farnesol, and its oxidation product, the aldehyde farnesyl. And Schmalek showed that if the stability of these compounds was increased, as in farnesyl acetone, or in the very convenient one, the methyl ether of farnesyl methyl ether, the activity was greatly increased, apparently, partly by making these substances more stable. A year or so ago, Law, working in Williams' laboratory, showed that chlorinated derivatives of farnesoic acid, methyl ester, in fact, of farnesoic acid, were even more active. Well, the chemical nature of the juvenile hormone, the natural juvenile hormone, may well vary somewhat from one species of insect to another. But just earlier this year, the active substance from the Cecropia silkworm has been recognized by Roller as the this one, you see, is farnesyl methyl ether, and the natural substance for Cecropia is this substance, the methyl ester of 7 ethyl 3,11 dimethyl 10 epoxy tridecadiene carboxylic acid. Well, in general, you can see, apart from this epoxy formation and the presence of an ethyl group here instead of a methyl group, the general form of this molecule is very similar to the farnesol derivatives studied by Schmalek. And all these substances produce effects identical with those given by an implanted corpus alatum. Now, the point of interest in the present context is that these farnesol derivatives in minute doses will reproduce all the morphogenetic effects of the juvenile hormone. That, of course, is because all the juvenile hormone is doing is switching the gene system and thus controlling the manifestation of one or other of its inborn or preformed characters. That is the link which Swammerdam had not got. But when it comes to saying exactly how the hormone exerts this discriminative effect, we are not in a position to give a clear-cut answer. We may summarize our conception of embryonic growth in the insect by saying that when the daughter cells of the zygote are exposed to different chemical environments in the egg, they are induced to follow special lines of development leading to the formation of the different parts of the body, the head, the thorax, the abdomen, the legs, the wings, the genitalia, and so on, and that each of these parts of the body can appear in a larval form, a pupal form, and an adult form, a larval form, a pupal form, and an adult form. And we know that these differences between the larva, the pupa, and the adult are controlled by different concentrations of the juvenile hormone. Metamorphosis is clearly an example of gene switching brought about by a controlling hormone. And there is increasing evidence to suggest that differentiation at the tissue level is likewise brought about by gene switching due to chemical differences in the cytoplasm to which the chromosomes are exposed. And there is yet another type of polymorphism which is controlled in a similar way. I refer, of course, to those examples of environmental polymorphism in which there are striking differences in form between different individuals of a species, although their genetic constitution is believed to be identical. Environmental factors of many kinds are responsible for determining which form a given individual shall take. But it's interesting to note that circulating hormones are very commonly the agents that are used to bring about this switch. And in some cases, such as the change among termites from the worker larva to the soldier, that is one, and the other one is in the aphid, the switch between the winged form of the adult and the wingless form of adult, which results from crowding or spacing. Both these switches, in both these switches, it seems to be the juvenile hormone itself which is used as the hormonal agent to regulate the change. The preformation that Swaminathan postulated has thus been driven back into the gene system. The next step will be to describe exactly how these genetic switches are brought about. We already have some highly suggestive ideas developed by Jacob and Monod and others to account for the adaptive synthesis of enzymes in bacteria that can be induced by the substrates in the environment. Perhaps these ideas will eventually provide the mechanism we are seeking. Well, in conclusion, I must return to the theory of Aristotle as developed by Harvey, that the insect larva is a walking embryo which builds up reserves of nutriment before it reverts once more to the egg stage or pupa and completes its development to the adult. This theory gave a satisfying interpretation of metamorphosis which held the field for 200 years after Harvey's day. But close study of the caterpillar of the butterfly or the maggot of the fly at once reveals that the resemblance between these creatures and embryos is only superficial. These larvae are, in fact, highly complex organisms, each closely adapted to the environment in which it lives. It is this adaptation which provides the key to the problem. It was pointed out by Darwin that when a larva feeds on different foods, lives in a different environment from the adult, it will be subject to quite different selection pressures. And its characters will evolve quite independently from those of the adult. the greater the difference in the way of life of the two stages, the wider will the structural differences become, until an intermediate pupal stage must likewise be evolved to bridge the gap between them. In some degree, those elements of the gene system that determine the characters of the larva are independent of the elements that control the characters of the adult. The light, please. In the end, we seem to have 2 or 3 separate forms locked up in the same animal. It was Sir Thomas Browne, writing of the silkworm 300 years ago, who expressed his amazement at the existence of 2 souls in these little bodies. There's nothing here that is new in principle from what we are familiar with in mammalian physiology. We accept the idea that male and female characters are latent in every embryo. The balance of genes and the endocrine constitution of the individual dictate which of these latent forms shall become evident. What is peculiar about the insect is the spectacular nature of the changes in form, the clear-cut character of the hormonal regulation of these changes, and the fact that the system is so arranged that the forms that are latent in the embryo are regularly manifested in succession during the life of the individual. The whole story illustrates the point that Malpighi and Swammerdam were tried to bring home to their contemporaries, that one of the most hopeful ways of advancing knowledge of the physiology of man is to study the workings of the body of the insect. Thank you.